This is the second part of today's session on imaging and lesions, and we will be talking about the bold signal based on which fMRI can be measured. There are two different ways of imaging the brain. We can use structural imaging, so making a still picture of the brain or body part using CT, PET, MRI, and this can be used to study the brain structures themselves, but also diagnose diseases and injuries. So even though this is structural imaging, there we can also measure changes across time, but on a relatively large scale. So for example, when people become taxi drivers in London, their hippocampus enlarges. So this is something we can measure, but of course we cannot measure very dynamic fluctuations such as cognitive processes. We would need functional imaging for that, because functional imaging allows us to dynamically map the moment-to-moment activity of the brain, and this can be done, for example, using fMRI. So we're looking at relatively variable processes here. All hemodynamic methods are an indirect measure of brain activity. So we're not measuring the neural activity itself, we're measuring something that responds to the brain activity. For example, in positron emission tomography, PET, we measure how we can, for example, use neurotransmitters that are radioactive or radioactive glucose, certain drugs, and so on. When we give those to participants or patients, then we can put them in a scanner, and the idea is really the more of the reactive agent is metabolized, the more of a radioactive trace you will see. But again, this is not measuring the neural activity itself, it's really just measuring the metabolization related to increased or decreased brain activity. And because we're using radioactive agents, this is an invasive method. fMRI, functional magnetic resonance imaging, measures changes in the oxygen consumption. So here the idea is the more brain activity we have, the more oxygen is used. And this is non-invasive because we don't need any radioactive agent. PET and MRI can be combined to get both the advantages of PET and MRI. And then we have, for example, a high spatial resolution and also a high molecular sensitivity at the same time, as shown here. The basic logic about the bold signal is that the more neural activity we have, the more oxygen is consumed. Now the brain consumes 20% of the body's oxygen uptake, which is a lot given its small size. And hemoglobin is a molecule in the blood that can transport oxygen. Now when neurons consume oxygen, they convert oxyhemoglobin to deoxyhemoglobin. This is what happens here. And then when neurons in a particular brain region, like here, are active, there's more oxyhemoglobin in that region because the neurons need to be, they need the oxygen to consume to produce the neural activity. Now, importantly, deoxyhemoglobin has strong paramagnetic properties which introduce local magnetic field distortions, shown here. So these blue molecules here, that's deoxyhemoglobin, and the black lines here are indicating the magnetic field, and as you can see, there's a distortion in magnetic field, and as a result, the protons are not very homogeneously oriented, but rather heterogeneously oriented. So we have a decreased homogeneity, and if you remember from the first part of today's session, the more heterogeneous something is, the faster dephasing you would observe, which then results in a faster decaying T2 signal. So the strength of the signal really depends on the concentration of deoxyhemoglobin in the blood. The more deoxyhemoglobin you have, the lower the fMRI signal. And the fMRI signal measured in this way is called the BOLD signal, the blood oxygen level dependent signal. The way that BOLD signals evolve over time in response to an increase in neural activity is called the hemodynamic response function, or HRF. This looks like this. We have an initial dip, so the BOLD signal decreases, then increases and decreases again. How do we come to this shape of the function? This can be seen here. So in the basic state we have some oxygenated hemoglobin and deoxygenated hemoglobin, and then at the onset of the neural activity we see that the oxygen is consumed, so that means that deoxyhemoglobin is increased, and then as a response to that the BOLD signal decreases. This is the initial dip. Less oxygen, BOLD signal decreases. Then this is overcompensated by allowing a lot of oxygen into that region, so the local blood flow increases, deoxyhemoglobin decreases as a response to that, and that means that the BOLD signal increases. So fMRI is only an indirect measure of the neural activity, because it doesn't measure the activity in the neuron, like for example we would do with the single cell recording technique. Instead it measures the metabolic demands in the oxygen consumption of active neurons here in this vessel, for example, in this blood vessel. And of course this raises the question, how well does the BOLD signal actually reflect the activity of neurons? And the answer is pretty well, as shown in this study by Lugotetis et al., where they simultaneously recorded intracortical activity, neural signals, and fMRI. And as you can see here, this is the localization. This is where they had the intercortical recordings, and they did use fMRI to image that region as well. So we're looking at the same region, and as you can see, there is the same activity pattern. The orange signal here is the fMRI signal, the BOLD signal, and the black bits here are individual spikes of the neurons that are shown with the intracortical recordings. So even though there's some delay, as you can see here, they correspond relatively well. So fMRI seems to be a valid way to measure neural activity. So to summarize, fMRI measures the ratio of oxygenated to deoxygenated hemoglobin in blood vessels of the brain, which makes it an indirect measure of neural activity. The signal measured in fMRI is called BOLD, blood oxygen level dependent signal, and the change in the BOLD signal over time follows the HRF. First, an initial dip. This is when oxygen is consumed. Then, as an overcompensation to the lack of oxygen, there's an increase of oxygenated hemoglobin and decrease of deoxygenated hemoglobin, which means the BOLD signal increases. And this is the three phases. It's initial dip, the overcompensation, and then some kind of undershoot here. The hemodynamic changes are relatively small. It's only 1 to 3%, for example, for a 1.5 Tesla magnet. And the hemodynamic changes are, however, relatively stable for the same participant in the same region. But the hemodynamic changes are relatively variable for the same participant in different regions or for different participants in the same region. The temporal resolution is relatively low because the BOLD signal peaks 4 to 6 seconds after the neural activity starts. The spatial resolution is relatively high, So a voxel has a size of normally 2 to 5 millimeters, and if you increase the magnetic field, if, for example, in 7 Tesla scanner, you can go lower than 1 millimeter. And by voxel, I mean the smallest addressable element in 3D. So this is like the resolution of fMRI. Just like a pixel in 2D, a voxel in 3D describes the smallest addressable element. And as you can see here, by increasing the magnetic field, you can actually increase the spatial resolution of fMRI. So much about the pulse signal and fMRI. Next, we will talk about fMRI data pre-processing.